no pictures and too many words. Okay. Besides, let's get back to the point. So what we're looking at are viruses. So viruses are just some kind of pathogen that would then infect pretty much any other living cell. So viruses technically aren't living. What they are is just special strands of either RNA or DNA that can infect normal living cells and force them to force them to replicate the virus DNA or RNA outwards, so that it can then spread and infect everything else. Okay, so it can kind of propagate without actually living. Viruses have a couple different structural features. So we already mentioned one, we're looking at RNA or DNA to kind of exist, and that will be localized in a central, what do we got, a capsid or something? Yeah. Okay. Your capsid is your very center part where you've got your DNA and RNA kind of stored in a protective cell or a protective section. Then you've got an outer section that goes through and holds that all in one nice little protective section. And then we go even further out to a membrane envelope that protects everything on the outside. So the idea is that we want to make sure that the virus wants to make sure that that RNA and DNA don't get destroyed by natural cell processes. So we're trying to protect that as much as possible outside the cells. And then once the virus actually infects, it can then inject that stuff into the cell, and in theory it'll be a little bit more stable and able to propagate. So that last part is how would we get it injected into the cell. What we'll find on the outside of your membrane envelope are these things that they're calling protein spikes. Really what those are a way to recognize a cell and then be able to inject, almost like a hypodermic needle, inject that DNA into the cell. Okay? So we've got these individual components to it, and then we're going to look at varying stages of how it actually then propagates through the rest of that. Okay, so we could look at a nice pretty picture. If I'd been planning, I could have shrunk the picture down and put the text around it. We could have talked about those individual sections. Okay? So we've got our RNA or DNA in the middle, which they're listing as some kind of nucleic acid. That's why it could be RNA or DNA. You've got your capsid kind of protecting it, and then you can shift outwards, and you've got your membrane envelope protecting it even further. And then you've got the spikes, which will act both as a recognition site and as a way to inject your capsid into the DNA or into the cell. Okay? So those spikes will identify an individual cell. So what we can look at is your virus coming up to an active cell. The spikes would grab a hold of the cell and then allow the passage of the genetic material into the cell. That genetic material then needs to get to the point where it can actually be passed on. Okay, so where is that going to happen? Is that where your genetic material is passed on the surface? Okay. Depending on which branch of life we're looking at, be it prokaryotes or eukaryotes, we might push all the way to the nucleus, or at very least the nucleosome. I always get that one messed up. Nucleosome is prokaryotes, right? Okay. So what? Nucleoid, whatever. Something with an N. Okay. We need to get that genetic material to that point, and then we need to be able to replicate that genetic material. Okay. Problem with that replication process. Our cell is going to replicate what? Its own DNA. So why on earth would it start to then replicate some other random piece of DNA that floats in there? And in fact, there's a lot of other enzymes that facilitate the cleavage of random DNA floating around so that we could actually recycle it. Okay, so somehow or another, the virus has to be able to get its DNA all the way into the nucleus and then somehow convince the nucleus to start processing its DNA. Okay. What's going to be the easiest way to convince the nucleus to start processing it? If it looks like the host's DNA. Okay, well, what's the easiest way to look like the host DNA? What's that? Do what?
To have it.
I don't understand what you mean by to have it.
Well, there are some viruses that inject their own DNA into the host DNA, like viruses.
So what our virus is going to need to do is get that genetic code actually embedded into the host DNA. So not just being in the nucleus where it can potentially replicate, but literally take that strand of information and make it one and the same part of the cell DNA. If it's now part of one and the same of that cell DNA, when it goes through to replicate, it's also going to replicate the viral DNA. Okay? When it goes through to transcribe its host DNA, it's also going to transcribe the viral DNA. Okay? So it's kind of an interesting process because it has to be able to infect, bring in a foreign species, and not only just get it on the surface, but then be able to transport it through the cytoplasm with all sorts of other things getting in the way that could potentially degrade it, then get it into the nucleus, and then get it beyond the nucleus and into the DNA system, where our DNA system has a very good high-fidelity replication system that actually prevents this kind of process from happening. Okay? So viruses are going to have a lot of extra kind of help along the way. That help is going to have to be something that they've got coded in their genetic information. Right? So when they get injected, they can actually push this whole process through. Okay? Another picture of the attachments. So what we're looking at is our spike interacting with a particular probably saccharide on the surface of our cells. Once it does that interaction, it'll pick up another co-receptor and it can then launch its little spike inside it, split open the cell membrane, and actually allow for the passage of the DNA through. Right? Sounds like a pretty violent process. Actually might have been interesting to look at, but I wasn't interested in that part of it, so I skipped that. Right? What's that?
Micro, just, well, we were on micro, so we were learning about viruses, and it's intense.
Oh, it is exceptionally intense. I just wasn't interested in that part of it. The retroviruses have certain components that are really common between them. Okay? So they need to be able to code for the coat on everything that's on the outside and get those spikes and that system set up. They also need to code for all their kind of protective processes all the way in and code for their DNA. And they also need, depending on what the genetic information is, some kind of way to get their genetic information embedded into the host system. A lot of your viruses have an RNA genetic code as opposed to DNA. What's the problem with that? To incorporate that genetic information into the DNA, we actually have to go from RNA to DNA, which means we need a reverse transcriptase. Okay? We're running the opposite of our central dogma, where we go from DNA to RNA to proteins. Okay? So we need the reverse process to happen. Also thought about looking at that a little bit closer, but I still wasn't all that interested in that. The one thing that I thought was kind of bizarre that the textbook did not mention is, to me, the most important part is that DNA, once we now have gone from our RNA to our DNA, what do we need to get that DNA to do? How does it replicate, though?
It has to use either the cell's machinery or they bring in their own.
So we need to somehow get the cell machinery to help us out. How can we get the cell machinery to help us out? That viral DNA needs to be in the host DNA. How does that happen? We're going to have to break the host DNA to inject the new DNA. Okay, so we're taking double-stranded DNA that was already nice and stable, that at no point do we ever really break except for the replication process. Okay, and even then, we aren't breaking. We're keeping usually a single strand continuous all the way through, and we're sliding in individual pieces. So for us to incorporate this new viral DNA, we actually have to completely break open the DNA strand all the way through. Okay? Sounds kind of difficult to me. So I pushed that a little bit further, and what we're looking at is something known as an integrase. Okay? The integrase is what's involved with that process. Okay? Again, not what our textbook mentioned. So our steps of infection, we have to inject, we have to reverse transcriptase to get our RNA into our DNA. So that's going to take our single-stranded RNA genetic information and turn it into our double-stranded DNA. Once we have the double-stranded DNA, we then have to be able to integrate it into the host DNA. Okay? That insertion process I thought was kind of neat. Okay? That process is facilitated by an integrase. Where does that integrase come from? The virus. The virus has to bring that in. Okay? So we'll go through the reverse transcription to generate our DNA. That is actually going to happen out in the cytoplasm, at least based on what I was seeing. Okay? The integrase will then bind that double-stranded DNA. Okay? And that's what's forming what's known as the intra or intosome. I may have left off the R. I was tired. Okay? That complex then gets chaperoned through the cell and into the nucleus. Once it's in the nucleus, it cleaves off a couple nucleotides off the 3' end, and then somehow it's magically covalently incorporated into the host. Okay? That magical incorporation I wanted to read more about, and I didn't find anything in the couple hours that I spent looking at this. Okay? But what I did find was looking at that integrase. Okay? So that's where I kind of focused my talk. Within that integrase, we have a couple important sections within it. We have an N-terminal HHCC zinc binding domain. What is that? What is the HHCC zinc binding domain? Nope. Hydrogen, carbon, carbon isn't much of a bonding interaction. Histidine, cysteine, zinc binding protein, also known as it's a zinc finger. Okay? So at the N terminus, we have a zinc finger, or a couple zinc fingers. Why would we need that? What do the zinc fingers do? So you're right, it does keep a shape. Binding motif to DNA. Okay? So it's a structural fold of our integrase protein that's going to allow it to hold on to DNA. How many strands of DNA does it need to hold on to, though? It needs 2, because we've got the viral DNA and the host DNA. So it has to be able to bring both of those near each other. So we're going to need a couple different binding motifs. Okay, so we've got the N-terminal end that has this zinc finger. Okay. I did do a little bit more digging. Would have been nice if I'd done this digging actually earlier, because then we could have seen how the zinc finger interacts. I was hoping that we could pull up a PDB image of this, of the zinc finger actually wrapped. But what we can see is the helix, and then our turn back into a beta sheet. The green atoms are our zincs. Okay? So that helix shape, the alpha helix, if you remember from the helix turn helix motif, that what ends up happening is those fit very nicely into the grooves of our DNA. Okay? So that's how it's actually wrapping and holding itself within the DNA structure. Okay? We're going to need a catalytic core, so some place to allow for that reaction to occur. We'll talk about that in just a second. And then at the C-terminal end of our integrase protein, we have another DNA binding section. Okay? That other DNA binding section is to bring in the other strand of DNA so that we can actually get the integration. Okay? That binding motif is known as, the SH3 motif. Okay? What we're looking at within the SH3, might be nice to know what that is. That's the motif shown in the upper right-hand corner. That is the SH3 binding motif. Okay? Couldn't find one with wrapped DNA on it, but if we take a look at that, that does look rather helical in shape, and I would guess what's going to end up happening is your DNA is going to thread in between that. So you're probably looking at those, looks almost like a beta barrel. So beta sheets wrapping around or fitting into the grooves of your DNA strands. Okay? So that's going to hold your DNA. And if we take a look at the C-terminal end of our integrase, we've got 2 SH3 binding motifs that will end up picking up our other strand of DNA. One big question that I definitely had was, if this is the whole protein, where did the zinc fingers go? Well, I don't know either. Couldn't find an image that I could 3-dimensionally rotate to be able to track that structure down, unfortunately. I'm going to guess it's somewhere in that big massive tangle. I did find a PDB image, but it didn't have DNA in it, and it made it a lot more difficult to actually track down how to fit it in there. So what's going on with the catalytic core? Okay, so what we're looking at is trying to cleave that DNA strand open. Okay, so we're looking at different types of integrases. And depending on the different type, I think there's actually a third one in here. I think HIV is running under a different type of integrase. Okay, but we've got two other types. You've got a serine integrase and a tyrosine integrase. Okay, why would we call them serine and tyrosine integrases?
Because it uses that amino acid to...
What we're looking at within those types of integration or integrases is that it's either a serine nucleotide amino acid or a tyrosine amino acid that's actually involved in the catalytic exchanging of your bonds. What do both serine and tyrosine have in common? They both have an OH functionality. Why would that be important? What can that oxygen do? Could be deprotonated and reprotonated. Neither of them, well, the tyrosine is slightly acidic because it's a phenol. Serine is straight up alcohol, so it's not going to work very well as an acid base. We do have the lone pairs on those oxygens, which means the lone pairs can act as nucleophiles. So when we're looking at our serine and our tyrosine integrases, what we're probably looking at is that oxygen acting as a nucleophile. What is it going to react with? The phosphate. The phosphorus electrophile of the DNA backbone. Okay? So if we take one of our nucleophilic oxygens, let's draw one over here. Whoops, come on, hydrogen. We can take that electron density and go through and attack that phosphorus. So now the question is, which bond breaks? So if we push in there, officially, if we're going to do organic chemistry, and I know this picture isn't the greatest, we break the pi bond on our phosphocarbonyl. I don't know what to call that. Okay. That's not referring to the carbonyl, though. That's the whole thing. We'll bring the pi bond back down, but now we have to kick out a leaving group. What leaving group do we kick out? Oxygen's a good answer. Which oxygen? We can either kick out the one that we were already, or that we just brought in from our amino acid, or we could look at breaking our backbone. Which one of those bonds is going to break?
Probably the one with the sugar.
Okay. Well, they're both attached to a sugar, so I don't quite accept that answer.
The one that has the last carbon.
How is DNA synthesized?
5'.
5' to 3'. So which oxygen do we want to actually be active to pick up our viral DNA?
3.
We want the 5' end. Okay? And I always get that backwards, so I might be wrong on that. Okay, so that bond cleaves, and what we'll end up with is a free 5' end. What can that 5' end do? It can then attack the 3' phosphate. No, it's got to go the other way around. Because the phosphate's on the 5'. Yeah. It's got to break this way. So our 3' of our viral DNA can now attack the phosphorus of our 5' host DNA to kick out our protein. Okay, our serine or tyrosine structure. We now have one strand of our DNA, of our host DNA, now linked to the other strand, or the strand of our viral DNA. What else are we going to have to do? We got 3 more points where we've got to complete this process. We now have to also be able to, 3 more points? 3 more may be a bit excessive on that. Because we're going to have to bring in, no, 3 more, because we're going to have to connect the other end of our viral DNA to the tail end of this guy. okay, the top here, and we're also going to have to do that cleavage on the opposite side to insert our DNA. Okay? You ask me, that's kind of a neat process. The exact mechanism for that, like I said, in the 2 hours that I looked at, didn't actually find it. Okay? What they're talking about is that we're looking at that serine and the tyrosine being critical to the overall reaction of the integrase, it actually functioning properly. So if it's critical to it, it's probably involved in our rate limiting step. It's somehow going to be contributing to this overall process. Okay? This seems to make sense. We can cleave that backbone, put it onto our protein, allow the other guy to come in, okay? And then we can go through and swap it around. Okay? Kind of a neat process. I don't know. To me, that was cool. Okay? Oh. Dang it. I animated and removed those so that I could draw it cleaner. So much for that. If we look at the viral genome, we've got the coat proteins, your reverse transcriptase, and the envelope protein. And then for a couple other types of viruses, you've got other things that are also being coded as well. Okay? So they've listed HIV and the Roux sarcoma virus as well. Okay? So those things are all part of that gene sequence and need to get coded across. If they don't get coded, we're not going to have the protective coating around the DNA that comes out of it, and if it can't protect that DNA, it's probably just going to get cleaved and it's not going to be able to propagate and continue to infect. Okay? So exam questions, I threw them all up there. Let's see, what did I do? 4 multiple choice and 1 true false. Ooh, I didn't answer the true false one.